Last time we left off on carbohydrates, so today we're going to go into another type of biological macromolecule, and it's called lipids. So the monomers of this class are fatty acids and glycerol. And these monomers come together to form polymer, so a larger molecule of multiple subunits, and it's called a triglyceride. And now let's delve into what each one of these monomers look like. So a fatty acid is formed of a long carbon chain, much longer than the one I'm showing you, so keep that in mind. And it's surrounded by hydrogens. And at the end here, there's a carboxyl group. That's a functional group. You'd probably want to just keep that in your memory for biochem. And I'll also show you a glycerol. So this is a glycerol. And for the purposes today, in this video, I'm going to have to extend this, so bear with me. Okay, and let's see how this forms a triglyceride. So I'll copy the fatty acid over. And I'll make another copy of this drawing, so I'm going to be able to show you a step-by-step. So what happens here is I'm going to circle these in red. This alcohol group or hydroxyl group and this hydrogen are going to break off, and they're going to form an H2O. Now, when these two break off, so I'm going to erase this and then move them elsewhere. So they form an H2O over here, and I'll move the molecule over closer to glycerol. And I'll have to move this double bonded oxygen up. This oxygen is going to be connecting the two carbons, so it's going to form an oxygen bridge. And if you notice, this is very similar to the one we did yesterday with carbohydrates bonding together, and this too is dehydration synthesis. So you can see here how the molecules together lose H2O, so they lose water, and together they bond together to synthesize a larger molecule. Now, this reaction takes place 3 times for the glycerol, so we are going to end up with 3 H2Os, and I will copy the fatty acid over to show you the rest of the places as bonded. Okay. And since there's 3 spots, now you might be able to understand why it's called a triglyceride, because you have 3 fatty acids bonded to 1 glycerol. Now, one of the interesting properties about lipids, such as this triglyceride, so I'll label this as a triglyceride, is that it's not soluble in water, so it's insoluble. And the reason why that is because carbons are all bonded to hydrogens. And one of the things you learn is that water is polar. Water is polar, and polar things can only dissolve polar things. And how do we determine polarity? Well, polarity is determined by electronegativity difference. So electronegativity difference is how much an atom wants to hold onto an electron. And if the difference, it's normally quantified numerically. If the difference is greater than 0.4, it's polar. And if it's less than 0.4, it's nonpolar. And hydrogen and carbon here, which you see are bonded together, they have electronegativity difference of less than 0.4. So it's nonpolar. And for that reason, lipids are not soluble in water. And, well, let's talk about the uses for lipids. Lipids are used for long-term energy storage. They're also used for padding and insulation. So think of, I guess, a seal. They have a lot of blubber, which are fats, to keep them warm. You also have steroids. Now, just to make this clear, steroid is not chemically similar to the one we just did. So it doesn't really look like a triglyceride. Now I'll show you a picture of a steroid. You should be able to identify a steroid. It has a pretty unique structure. So steroids typically have a four-ring structure. Three of them are six-sided, so you see three six-sided. And you also have one that's five-sided. And steroids can do a variety of things. They can act as hormones, so developmental hormones. However, let me make this clear, not all hormones are steroids. And a prominent example is insulin. Insulin is a hormone, but it turns out, bear with me for a second as my computer is having issues. Insulin, it turns out to be a protein. So not all hormones are steroids, some of them can be proteins. And yeah, compare this structure to the triglyceride. And you'd be right to think that, wow, these two hardly look similar at all. And the only reason why steroids are classified as lipids is because they too, like these triglycerides, are both insoluble in water. Now let's go back to the triglyceride and we're going to talk about saturated versus unsaturated fats. You've probably heard of these when looking at your food at the nutritional breakdown. So, difference between a saturated and an unsaturated fat is, well, this is a saturated fat. And a saturated fat means that all available slots for hydrogen are filled up by hydrogen. So it has a max amount of hydrogens. In other words, it's saturated by hydrogens. And might lead you to conclude, rightfully so, that unsaturated means that it has less than the maximum amount of hydrogen, and that happens when you have a carbon that's double bonded to another carbon, and that leads to spaces that where hydrogen could be, there isn't. Now, normally it doesn't look like this, so if you have an unsaturated fatty acid, it tends to be bent. So you'd see something like this. And the reason is because it's unsaturated, it puts a kink into the structure due to the carbon's double bond. Okay, and finally we'll go through one last important function for lipids, and that's as a cell membrane. Now, cell membranes are composed of things called phospholipids. Our cell membranes are called phospholipid bilayers, so they are composed of phospholipids. Sorry, I'm having some issues with my stylus. So all that means is in a triglyceride, you have a structure like this. You have the glycerol connected to the 3 fatty acids. And in a phospholipid, one of these fatty acids, so let's say this one, instead of being a fatty acid, is replaced by a phosphate group. And that results in a phosphate group. Let's just pretend that's a phosphate group. And really key implication for this is that phosphates are soluble in water. So this is soluble. However, the rest of it isn't soluble, so the rest of this is insoluble. And now I'll show you a better picture of a phospholipid. So this is what a phospholipid looks like. You can see that here has a bulb thing, which is just a phosphate group. And what happens, because this thing is soluble in water, while these aren't, so these are insoluble, you end up with a molecule that's partially soluble. Half of it is soluble, half of it is insoluble. And the implication is that this part is going to be hydrophilic, or rather water-loving, which is just another word for soluble. And this part is hydrophobic. And this allows these molecules to form a cell membrane because one side is hydrophilic and the other is hydrophobic, so it manages to keep the inside different from the outside by separating it. And yeah, if you stick a lot of these phospholipids together, they will just form a cell membrane. And that will be all for lipids.